everything's working. Right, yes, so pleasure back here again at EVA. I think a bit of a regular sort of like place to film my stuff, which has been a sort of real pleasure. Sort of, I feel it's like a friendly community. So what I'm going to be talking about today is it's a work in progress of almost like a project I've been working on for, I guess, just under a year now. So this is, and basically it's going to be quite practical stuff about just like what this project is. The project is actually, we've got the current prototype of it basically up in the communal area. So we're going to do talk about things with slides, and then we can go out there and I'll do a bit of a demo around the actual installation thing, and people can try it out. And I'm also just really interested in feedback, because they're saying this is still a work in progress. In particular, we're working out things like what are the right sort of controls, what are the right things that people want to interact with this, using, and does this, what questions does it raise? Because actually it's meant to be almost like a piece to provoke discussion and debate about sort of some science sort of issues. So I guess first of all, the title, I guess, as far as EVA is concerned, is Twitchy Cells. I'm sort of wanting to change it a little bit to Synchrony, which has become the sort of new approved title together with the researchers I'm working with, which maybe is a bit more meaningful about what this project's about. So this is a sort of collaboration project. So basically it's together with a research group over in Oxford called the Srinivas Group, basically named after Professor Shankar Srinivas, who is the first picture you see there. So basically they're a research group doing really almost like fundamental developmental biology research, but in medical research institutes. They're in the Institute of Developmental and Regenerative Medicine over in Oxford. So you see the controls introduced, that's Shankar, and then Shifa, Tomoko, and Jonathan. So basically that is the Srinivas group. And basically, I guess Shankar saw actually the work that I was showing at EVA last year, which is this thing called digital chemotaxis. That's all about social amoebae and the rules they use to sort of like coordinate behaviour and things like that, which I was presenting inside a Victorian stereo viewer. And I guess actually just shortly before EVA last year, there was a symposium over in Oxford, and I was showing the work there. She saw that and then basically sent me an email going, hey, talk to us, you might be interested in doing a project with us. So given that my work is generally about being inspired by biology ideas, particularly almost developmental biology, and but trying to use it as almost like a creative medium, almost like to make these sort of rich, unruly, complex systems, which are hopefully interesting to create with. Having genuine, people are doing fundamental research in this area, it's like, okay, that's really cool. So basically the area of study is patterning and morphogenesis of the early embryo. Basically, how does embryos get the structures and shapes they do through their processes of development? Which, if you know my work is, yeah, you got me already. That's sort of like, what I'm sort of interested in. In particular, this project that I'm working on, what they're actually looking at is rhythmic contractions rather than just overall structure, which is a bit of a change for me. So they're basically particularly good at the early embryos. These are 7.5 to 8 day old embryos. How they start to have this thing where it goes from being just like a few cells, just like contracting almost like randomly. And it actually becomes almost this much more sort of structured movement. So this video that you're seeing is cells just randomly triggered, which basically they're sort of called calcium ion concentrations. When the concentration of calcium ions gets to a certain level, the cell just goes and contracts and its neighbours towards it. And the idea is that this and then the big thing is that the cells can then influence their neighbours as well. It's like when they suddenly contract, they release calcium ions, which can then increase the concentration in their neighbours, so they can also trigger their neighbour as well, which is sort of how we start getting synchronisation. So after which is happening, to you get these waves of contractions happening in the embryo. Apparently it's the first structured movement that you see in a very early embryo development. So these are confocal microscopes of images with cells which are fluorescing. Things like with their calcium ion concentrations. Then transgenic embryos. And then gradually this develops into richer and richer, more complex structures. You get patterns of movement, and the movement sort of affects the growth of structures. Actually, that's one of the big questions until recently is really open. No one knew is it the movement happens first, or is it the structures happen first? Is it the complex structures affect movement, or is it complex movement affect structures? Some earlier work by this group, essentially, I think they now definitively say movement happens first. Movement, these patterns of waves, is what drives the creation of structures. And gradually, over the course of the embryo, becomes the heart. So it becomes this really rich, complex structure where the patterns of waves going through this structure is what makes the heart beat. So really fundamental one of the important science. And science directly to do with medicine things. They're actually talking about understanding that developmental process is potentially really important, particularly for some heart conditions, which heart is maybe developed slightly differently from how it's meant to. Fortunately, it's very close to home to me. Some of you know I've got what's called hypertrophic cardiomyopathy, which is basically one of the two main medical conditions that they're thinking this research could be very directly related to. Essentially, my heart has grown, my heart muscle has grown a little bit too thick in some areas, which means it can just, the patterns can be wrong. It's wrong, your heart doesn't beat, which is not a good idea. So maybe you can implant a defibrillator to shock my heart back into action when it goes wrong. So these people are doing deep fundamental science research, which is maybe directly personally relevant to me, I guess. So this is this project. So the project is synchrony. So it's very much inspired by the research, but not just trying to demonstrate their research. So we've got some other directions that we actually go as well. We seem to be getting on quite well as a group. This particular project, let's just explore this idea of cells that can contract and cells that can influence their neighbours to contract too. So it's like a sort of interesting system. What happens? What behaviours do we get? What kinds of movement do we get? Do we see things which are just like we see in the embryo or not? So I'm not trying to accurately replicate the embryo. It's more explore a bit wider what happens. Which I think is almost the one I said about the other projects. It's the exploring the range of possibilities of this system. So things we're sort of interested in is what sort of patterns of rhythmic motion do we get from that just simple system, cells that twitch, cells that can influence the neighbours to twitch. How do transitions from just individual cells randomly doing stuff independently to these sort of synchronised structured things sort of happen? See that happening. Okay. And one particular question that the researchers are really interested in, because it's almost like the directions, next steps their research. Consistent emission centres for the waves develop. You start seeing consistent things in the structure where waves keep on coming from the same place. They keep on coming from the same place. What sort of patterns are there? How they emit, things like that. So there's some questions that sort of think are relevant, potentially relevant to their research. That kind of this. So I guess it's what we're trying to make something which is hopefully useful for just like public engagement things. It's funded, I guess, under the public engagement funding and their research. So hopefully they can take it to science fairs and things like that, and it's a fun thing for kids to play on. But we actually seem to have some really quite nice, deep philosophical arguments about discussions about it, and then asking questions of their next research regulation on these sort of like emission centres and things like that. And it's like, okay, that's interesting. Yeah, I'm not sure how would we put this maybe into the model. What would that be? We're having to have a nice dialogue. They sort of like me freaking out there. I like rocking, talking about things. So that's each cell. It's a potential level. And the potential level, if it goes above this setting, that's what makes the cell contract. The cells contract, it can share that some potential, increase the potential in its neighbours a bit. Okay, so it can also hopefully trigger its neighbours to potentially contract as well. So simple, sort of, you see, closely inspired by what I was saying. Implementation-wise, it's C++ and CUDA. It uses optics for rendering, which is basically NVIDIA's library for writing your own ray tracer, but using the GPU. The original implementation was based quite closely on my old cellular forms code, but that's not designed to be interactive, so we got almost like a proof of concept thing working quite hard on my old cellular forms code. But then we sort of rewrote it in a modern sort of code base because we decided actually making this genuinely interactive. It's so that, the simulation happens 30, 60 frames a second. You can modify and change parameters in the simulation yourself and influence it. It starts to become interesting. So basically, saying the next sort of code base is quite just like things. And the other thing we put in there is sonification. So there's sound, audio produced directly from the simulation. Hopefully is meaningful. I think really helps immersion in the work. I mean, one thing I've really noticed is if people put the headphones on, if they don't put the headphones on, they often interact with it for about 30 seconds or so. If they put the headphones on, 10 minutes later, they're often still interacting with it. There's a level, it's less obvious stuff, but I think it really helps you understand patience with it. Often the work is too much just like you do a few things and then just leave it for a while and see how it develops. I think hearing audio changing, modifying, you understand that things are happening. You understand that the controls are actually doing something, even if it's not visually immediately obvious. Hopefully you hear it and you hear things in the audio. So I think the audio is a new thing. I'm mostly a visual artist and I'm naively learning more about the audio side of things. I think a work like this, I think it's really important. So essentially what I'm doing is 3 audio tracks. First 2 are basically taking these potential levels in every cell, contraction amount in each cell, and just mapping a histogram. So it's like let's create a chart of what the contraction levels are in each cell, and then use that just to drive different frequencies. So it's almost like every cell is emitting 2 different tones, one based on how the potential level currently is, which is like C sort of tone, and one which is based on how much it contracts. So it's not much more until it contracts. There's a slightly more mid-range frequency that comes from it having you're hearing the contracted cells almost. And then the third one is just like a Geiger counter. It's just like little clicks where every time a cell contracts, it creates a little click, which is just implemented as a simple physics-based sound synthesis system. Exactly like this. That's just spring, which just gets hit by a little impulse every time a cell contracts. There are 250,000 cells in the simulation, and what we can do, just like 250,000 cells, all just like hitting this little tune, and it works. It produces little crackly little sounds when you can hear the cells starting to contract. So that's the sound side of it. What was the other one? I've got this thing with Species Explorer, which is my present the artifacts, but it's part of my practice. It's almost like the system I use for doing evolutionary algorithms and machine learning. Just explore the parameter space, find the points where parameter combinations that things seem to do interesting stuff. So yet again, I'm using Species Explorer. So what I've done is I've found a number of different points in the parameter space that seem to sort of do what I think is interesting behaviour, which I can then almost give as presets for people to start with. So, a little bit about the results. So, lots of contraction. First thing is that we naively just implemented the things, as I described with the graph, didn't actually really work. what we actually found was either we just got little pop things or little sort of like little bits of movement that one cell would contract and maybe into a few cells that die out, you just get the thing, you didn't get the waves, these big waves of contraction happening. Or you just raise the level a little bit more and then suddenly it just almost became an out of control chain reaction explosion. You're seeing here that there's like a few contractions happen and then it goes, it just becomes out of control. So basically what we found is we could get some waves of activity, but we had to fine tune it like mad to get there. So we did like 1,000 samples, we had to find, when inducing waves of activity. So we found it and added just a little extra thing to the simulation, which is after every contraction there's almost like a recovery period. So we're saying that each cell, when it contracts, it can't immediately be influenced to contract again. There's a time when it becomes unresponsive to taking up the potential for its neighbours. And it just based on its contraction levels, only as it relaxes back to its original size, then it starts to be able to be influenced by its neighbours again. It works brilliant. So basically, we started going to fine tune like that to get waves patterns of activity. It just seemed to happen almost like we had just random waves and it never triggers, and then you get another set threshold and then everything starts producing waves. It's biologically justifiable. Okay, we got the so they're not aware of whether that's what really happens in cells or not. But yeah, it's really interesting. It's much plausible, something that they knew. So a little bit how I presented it. It's a formation on a stone stereoscope, which is this type of Victorian stereo viewer. So basically originally they were just like two mirrors, two pictures, and no lenses. Your eye is basically in the middle, and then one eye is seeing one picture because of one mirror, the other eye is seeing the other picture because of the other mirror. And I like it's just basically how high quality the quality of image circumstances for these Wheatstone viewers, I think are brilliant. And just the nice artifacts. It's great. You can see a bit more what's going on from the outside. So, this system, so it's two front surface mirrors, really nice high resolution screens. Fabrication of this, which is based on previous design. There's a thing like actually the EVA24 exhibition that a few people have mentioned in previous days. So I had a work in the EVA24, which was a Wheatstone stereo view a bit like this. This is a new iteration of that design. But designed to be mostly more robust, easier to assemble, option protective front screen, which we call the anti-finger pokery. So basically, you've got the option of having a full front rotation it's a full front screen to stop people poking their fingers on the front surface mirror. So they get fragile front surface mirrors. Very nice. Sort of like silverings on the front of the glass, which means a much clearer image when it's fragile. Okay. Anti-finger. Nice. Having this as a thing we can potentially give to the Oxford team can have and assemble onto themselves. So, testing, we did first session over in Oxford, talking about cells and things like that. So this was just with other medical researchers over at the campus. Then basically we get to people. We've got the second round of user testing is here. And what we call an interactive controls. But basically, there's two other things from the simulation control. So it's the potential levels in the cells and how much does the cell influence its neighbours when it twitches. So hopefully more characters from outputs. So if you just want to try and start from somewhere, I think it's sort of interesting things. It's audio, headphones, and it's out there. I have to see how many people can get. Okay.